Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing who will be taking part at the CanMed 2021 event this April 12th through 14th at the Pasadena Convention Center in Pasadena, California. I am your host, Ben Amirault. I'm the marketing manager at Medicinal Genomics and proud member of the team that puts on the CanMed conference every year. Head over to canmedevents.com to learn all about the CanMed 2021 event and get your tickets at our special early bird rate. While you're at canmedevents.com, be sure to sign up for email alerts to stay up to date with all the news surrounding this industry-leading event. We still have our safety keynote, panel discussions, and additional oral presenters to announce, along with some other surprises. And here's a special tip. If you sign up using the form on our podcast page, canmedevents.com/coffee talk, you will be entered to win 2 tickets to the CanMed VIP dinner. You will also find all the previous episodes of the CanMed Coffee Talk podcast there as well, so it's a good page to check out. This episode's guest is plant pathology expert Dr. Zamir Punja of Simon Fraser University's Department of Biological Sciences. Zamir's research investigates the causes and management of plant diseases on cannabis and hemp, including Fusarium, Penicillium, powdery mildew, botrytis, as well as other new and emerging plant pathogens. His CanMed 2018 presentation featured several amazing images Zamir's team captured using an electron microscope. The image of powdery mildew mycelia wrapped around a cannabis trichome is one I won't soon forget. Be sure to check out the link to that video in the show description. In addition to his work on plant diseases, Zamir's team has used their electron microscope to investigate the structure of cannabis trichomes and will present those findings at CanMed 2021. I expect we will see even more amazing images during that presentation. In fact, Zamir gave us a few sneak peek preview images that I've included in the show description. Topics discussed on the podcast include how diseases infect and affect cannabis plants, which cultivation methods are more prone to infection, how to breed for pathogen resistance, which pathogens pose a risk to consumers, and how each of the 4 observed cannabis trichome structures differ and what each might be responsible for. Before we get to my conversation with Zamir, I want to thank this episode's sponsor, the Growers Network. Growers Network is committed to providing the highest quality cannabis content, education, and resources. Be sure to check out their CannaCrib series, your behind-the-scenes look at some of the most sophisticated and technologically advanced grows in the world. Visit them at youtube.com/growersnetwork. And finally, this episode of the CanMed Coffee Talk podcast is fueled by the Hemp and Coffee Exchange. Hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, without any further ado, please enjoy my conversation with Dr. Zamir Punja. Good afternoon, Zamir. Thanks for joining us.
Oh, thanks, Ben. It's a pleasure to be here and talk to you.
Absolutely. And I'm really excited to talk with you today because I know you've done a lot of research into plant pathogens and diseases that affect the cannabis plant. And coincidentally, our team here at Medicinal Genomics has developed some qPCR DNA-based tests that can identify some of the very same species you've worked on. So although I'm familiar with some of the species and I've read about how they can affect the cannabis plant, we don't get to work with any actual plants here in our lab like you do. So I'm really interested in getting your perspective. And it's really fascinating to me. So with that, I think maybe a great place to start is with Fusarium, because I know that your team recently published two papers on that fungi. So I was wondering, can you tell us a bit about Fusarium and how it affects the cannabis plant?
Yeah, thanks. So we started working on these various diseases about 4 years ago. And the first thing we did was conduct surveys of a range of different producers out here primarily in British Columbia, although we did get a chance to get some samples from back east in Ontario. And the first disease that popped out, the one that was the most visible, was Fusarium. And on the plants, yeah, on flowering plants, Fusarium will show up as sort of a yellowing of the lower leaves, which is then followed by, in many cases, a wilting, and eventually, if the disease progresses, it'll kill the plant. So, in many other crops, it's referred to as Fusarium yellows. But what we saw in cannabis was more or less a root infection and a crown infection. And so, when you see a plant that's yellowing, that's wilting, if you examine the roots, they're going to be brown. Some of them may be rotted off. And then, if you look at the crown, you'll see sort of a pinkish orange, maybe even a dark brown discoloration that would indicate that it is Fusarium. So you see it on those flowering plants, but even more importantly, we found it on vegetatively propagated cuttings. So, a lot of the growers obviously start their plants from cuttings, and in Fusarium, we saw a lot of damping off. And so, in other words, rather than the cuttings going ahead and rooting, they were starting to literally fall over, and they rotted. And that came as a surprise to us. And so when we isolated from there, we found a lot of Fusarium. And going back to your earlier point, the main species is Fusarium oxysporum, which is the Fusarium that causes a lot of wilt in many other plants like tomatoes and basil and lettuce. In addition, we found Fusarium solanae. Solanae is the one that sort of causes more of the damping off and the root rot. And then the third species that we found just recently this past year was Fusarium proliferatum, and that one actually moves up the plant. It actually causes the crown infection, but then you start seeing discoloration pretty much all the way to the top. And so those three species you can sort of tell by looking whether it's a crown infection or root infection or whether it's progressing up the plant. But I think, as you mentioned, the PCR diagnostic tests are going to be the most accurate in telling which of those three species are actually present in a crop.
Now, you mentioned that in flowering, it can kind of present as having yellowing leaves. Are there signs and symptoms that can be visible sort of before that flowering stage?
The only symptom I would say is a stunting. So in other words, your plants haven't grown to the same height that they should have. They don't look like they're really happy. Maybe you think it might be a fertilizer problem. It ends
Mhm.
up that it's actually Fusarium that's infected the roots and potentially started infecting the crown.
Interesting. And you're saying that, cuttings can obviously take the if a mother plant is infected, can kind of take it into the cuttings. Would the mother plants really be showing any signs and symptoms, or is that sort of is that sort of the problem?
So that's where the problem actually started. The mother plants tend to not show any symptoms. And so we think the Fusarium originated, I don't know, many years ago, even before we started sampling 4 years ago, from mother plants or disease plants that were maybe passed along from one grower to another or from one friend to another, unknowingly that they already had some Fusarium probably in the roots or maybe in the crown. And then those mothers are grown up, and if they're really diseased, you will get the mothers dying. They will
Mm.
collapse, and obviously then it's time to get rid of them. But if you don't see those symptoms and you take cuts from them, you run the risk of actually transmitting Fusarium into the next crop.
And I suppose, and you can correct me if I'm wrong, but if you did have an otherwise healthy mother plant that develops the infection later on in life, you wouldn't really see that stunting on the initial growing phase. Is that fair to say?
Yes, correct. And it's hard to tell when a mother is well too old, if I can say that. We
Hmm.
tend to recommend mother plants be tossed out after 6 months to a year, and not keep them for 4 or 5 years, as some producers might be doing. At that stage, I think you might start seeing symptoms of yellowing and stunting, in which case it's time to get rid of them.
Right. And I know that it's not uncommon for a flowering plant to sort of, have some yellowing leaves as it sort of directs more nutrients into creating buds and things like that. So is there more of a telltale sign of what's kind of typical yellowing versus a Fusarium infection?
Yeah, so most of the infections that we do see that are quite severe would probably be within the first 2 weeks of flower. That's when the plant is producing, trying to get a lot of its energy into the flowering phase, and it puts a lot of stress on that particular plant, and that's when you start seeing the yellowing and the wilting, and in many cases, those plants actually die before they get into full flower. So when you got a
Wow.
plant that's in full flower and it may be showing a little bit of yellowing here and there, that could just be nutrition or environmental stress or something else. If it's fusarium, it's probably going to go down within the first 3 weeks of flower.
Wow. And now, is there anything you can do once you've identified a Fusarium infection?
Yeah, so the first place to start is with the cuttings. If you know your mother plants are clean, or if there's a way to test those, we actually haven't developed a testing method yet to test the mothers to see if they're carrying fusarium. And part of it is, the plants are large, and we don't know what part to actually sample for the analysis. But if you know the history of the mother plant, and it's a good source and it's clean, the first place to test would be the cuttings. So cuttings start dying, they're not rooting well. It could be fusarium present there. We're looking at a number of biological control agents that are registered. So, for example, trichoderma, gliocladium. There's products out there that are quite effective in keeping the fusarium on the cuttings at bay, provided they're added early. So, in other words, once the cuts are made, the cuts could either be dipped in fusarium, or sorry, in the trichoderma or the gliocladium, or you can apply a drench before the cuttings are actually rooted to actually prevent the infection from getting in.
And now, so how big or widespread is the Fusarium problem? Is it something that's pretty common in cannabis cultivation?
Yeah, so it's interesting. The short answer to that is the more we look, the more we find. I think I would have to say out of the 12 or 15 commercial producers I've sampled, every one of them had either very very trace amounts of fusarium to fusarium in about 30% of their plants. So I think it's there. It's had a chance to sort of move around. And depending on the variety or the strain, depending on the growing conditions, it may or may not be a major problem, or it could end up being a major problem in some grower facilities.
Yeah, and you bring up an interesting point. So, do environmental conditions kind of play a role in this? I know that, for example, with powdery mildew, if humidity is really high, that tends to breed more powdery mildew. Where if you have sort of your levels in check, it's less of an issue. Is there a similar kind of environmental trigger for Fusarium?
So fusarium, it grows over a pretty wide range, anywhere from 20 to 30 degrees Celsius, or you're looking at 75 to 90 degrees Fahrenheit. I would say overall that as it gets warmer, as you start hitting the 90 to 95-degree range, fusarium can be quite a bit worse than it would be at lower temperatures. It also seems to like moisture, and of course that's kind of hard to avoid because we tend to really water the plants and make sure the cuts are well watered. Fusarium will grow, and actually it'll spread with water, so you can have spores moving from let's say a propagation room where you've got cuttings into an adjoining room. If the water is being recirculated, you can actually get fusarium spores moving from one location to another.
Wow. And I know you mentioned that there could be some treatments that you do to the cuttings after you take them from the mother to sort of maybe prevent an infection. But so say you already are a few weeks into flowering and you're seeing those signs and it looks it doesn't look good. Is it a lost cause at that point, or is there anything you can do to sort of salvage that plant?
Yeah, no, I would go in and pull those plants out, particularly the ones that are yellowing and starting to wilt. There's always a concern that as the fusarium develops on the roots and the crowns, it's going to produce a lot of spores.
Mm.
And if you've got plants, let's say, sitting on the same table or in the same growing area, you could have spread, as I mentioned, in the water. But interestingly enough, it also moves in the air. So if you've got fans moving spores back and forth, you could actually be moving inoculum from that diseased plant or diseased plants into other areas in the growing room. So my recommendation: plants look bad, they're yellowing, grab them, take them out, bag them, and toss them.
Wow, that's interesting. So does that mean that like spores could even infect, maybe if there were some plants that were further in their development, they have full flowers, could it actually infect the flowers themselves?
Okay, so yeah, so that's a good news, bad news story. The good news is that if the spores do move around, they will not infect plants at flower or even at a younger stage. This fusarium seems to like to get in early,
Hmm.
so it'll get in with the cuttings. It might get in the vegetative stage, but once you've sort of passed that, the plants seem to be able to withstand the infection level. So if the plants have made it through at a certain stage, you're probably not going to worry about fusarium moving around too much from plant to plant. But we have found that if spores are produced, let's say there's a plant dying, and is producing spores at the base, at the crown. Those spores, once they get up into the air, can actually infect flowers of a neighboring plant. So if you've got plants a little bit further down, those spores can actually get up into the flower head and establish themselves in the flower if the humidity is right. If it's really humid and if it's hot. And so we've seen what we call fusarium flower rot or fusarium bud rot, which in many ways looks a lot like botrytis,
Hmm.
but it's only when you isolate and recover that you actually find out it's not botrytis and it's actually Fusarium.
Wow. And now, would that pose any health harm or health risk to consumers if a flower was infected with that?
Yeah, yeah, and you're probably familiar with Fusarium head blight in cereals and Fusarium in corn. We know many of these species, things like Fusarium oxysporum, can produce mycotoxins,
Hmm.
and there is a risk that if you had severe infections by Fusarium in the flowers, that could cause certain mycotoxins to accumulate. Because we've only just published those two papers in the last month or so, I think the regulatory agencies and I think in general, the community hasn't quite recognized that there is a potential for Fusarium species to infect the flower. Again, it's not something that I would necessarily raise a flag and say, this is the end of the cultivation of cannabis. I think it's it's there. I think we're going to find a little bit of it. I would say maybe 1% of the flower could have fusarium, but it's something that I think we need to be aware of, and certainly there should be screening methods for detecting some of the specific mycotoxins produced by fusarium.
Yeah, I mean, that's really interesting because I went back and I rewatched your lecture from CanMed 2018 where you're talking about a lot about fusarium and some of the other pathogens that can infect cannabis. And a thing that stood out to me is that you were mentioning that Penicillium, which is another species that can infect the plant, is sort of a similar thing where it can produce mycotoxins in the flower. And I know that regulatory agencies aren't specifically looking for Penicillium or Fusarium. Um, they'd be kind of covered under the total yeast and mold umbrella, but much like with Aspergillus, I mean, unless you're specifically looking for that, it can sort of get underneath that total threshold, and maybe actually be posing a risk to consumers. So that's a little concerning, and maybe something that regulatory agencies need to take another look at. Do you agree?
Yeah, so you guys at Medicinal Genomics were the first to actually show that some of these penicillium species, like Penicillium citrinum and others, are potential mycotoxin producers. I mean, and this was work that was published 3 or 4 years ago by Kevin and so on and his group, and that sort of alerted me to the fact that we should be testing our cannabis flower for penicillium as well. And sure enough, we found penicillium. It's penicillium is everywhere. It's found in soil. It's found on plant material that's starting to decompose. It's found in compost. It's found in people's homes. Like it's not something that you necessarily can kind of eliminate from the growing environment. But what tends to happen is when you've got a lot of it building up. So let's say you've got some plant material that's decaying, and producing a lot of spores, or the floors aren't necessarily kept very clean, or the filtering system or UV system isn't collecting all the spores. Those penicillium spores land on the flowers, and they can stay there. And after a while, they can start growing and producing more spores, which in turn can then maybe lead to certain mycotoxins that are produced by the Penicillium species. So I know Penicillium is a big component of the overall yeast and mold count. But as you
Mhm.
mentioned, all they do is count numbers, right? So once you're under a certain limit, you're good to go. But they could potentially be mycotoxins building up in there.
Yeah, no, that's and you mentioned sort of if you have plant material collecting, and it reminded me of that slide that you had showed of a plate that you had left out in a trim room, and just seeing the sheer amount of molds and things that grew on that. Um, that was pretty
Yeah,
pretty shocking to see.
I was shocked when I saw that. And just to clarify, there in the trim room, those flowers and those stems are really being agitated. They're either going through some mechanical destemming process, and that physical breakage of that plant material, I think, is releasing a lot of spores. And they're sort of like think of the cloud effect. Those
Mm,
clouds are sort of moving up into the air. So when we put those plates out, when the spores landed back down onto the plates, and we had them out for about an hour, they were just covered in penicillium spores. So, we're finding flowers that are dried, hang dried, and are not agitated to the same extent as buds that are pushed through mechanical destemmers and trimmers. That mechanical agitation is causing more spores to build up on product than if you actually hand dried those stems.
yeah, and I have to imagine that if you're an employee there, you're breathing all of that in, unless you're wearing a mask, and I'm not sure if they are. I would think that if you're not, that's something that you definitely want to consider.
Yeah, and then that's the first recommendation we made is let's get those workers to wear either N95 type masks, or masks that would have respiratory abilities to take those spores out so that their lungs are not exposed to. And it's not just the spores, it's the dust as well.
Right. Yeah, masks, they're becoming more and more common, so I wouldn't be surprised if folks
Yeah,
adopted that.
for sure.
And another thing that stood out from that presentation was you actually looked at whether certain mediums were actually contaminated, and you were finding that some coco coir that you had looked at was coming in contaminated. That was that was surprising to see as well.
Yeah, and so again, the cocoa core is nothing more than plant material, mostly fibrous plant material that's taken out of the cocoa husk and dried, and in some cases it's sterilized, and then it's sort of packaged in large bulks and shipped over from overseas countries. So there's always a chance that microbes and mold and other things can be carried over. Nothing in particular other than the penicillium and the aspergilluses and so on. But once you place that cocoa in a moist environment, so you're wetting it up and you're adding fertilizer and you're using it as a growing substrate, that allows the residual spores that are in there to start with to all of a sudden start multiplying. And so what we were seeing is that when you had those cocoa substrates, spores were being produced that we were catching on these petri dishes that we were laying out. And the concern was similar to the Fusarium concern, is that once they get up into the air, they could then land on these flowers that are
Mm,
maturing or getting ready to be harvested, and then you're picking up the spores that were building up as a result of the substrate that you were using to grow the plants in.
right. And that's not to say that all microbes are bad, right? So there are certainly beneficial microbes that may be present in sort of an organic medium like that as well, right?
Yeah, and so we've actually sampled recently a lot of organic substrates, and as you mentioned, the organic growers tend to use soil. And soil is good in the sense that it has a much better buffering capacity against pathogens than, let's say, hydroponics or rock wool. So on the positive side, we're seeing less disease develop in roots and crowns of plants that are grown in soil,
That's
because you've got
interesting.
so much of that buffering capacity. Um, but we're finding more microbes in the air in an organic production system. Again, going back to this idea that soil has a lot of beneficials and other microbes in there, more of them are getting into the air than you would find perhaps in a hydroponic or a system that uses rock wool. Um, the concern, the irony here is that even these beneficial microbes, let's say coming out of organic systems, if they do land on these flowers, they would be rated again as a general colony forming unit, and it
Yep.
may actually cause a lot of the product to fail. And as a general rule, and this is just sort of a broad statement, a lot of the organic growers are having problems passing their product, more so than the guys that are using hydroponic or rock wool, because of that background level of microbes, some of which are beneficial, getting onto the flower and causing the product to be rejected, because we don't discriminate right now between the good and the bad.
Right, and that's something that we see on our end too, because obviously we're involved in kind of testing the end product to make sure it doesn't have that contamination. Um, and yeah, we see cultivators that are using trichoderma, as you mentioned earlier, and these other beneficial microbes that might be testing hot for some of these total count tests. So I think it's great the work that you're doing and others are doing to sort of identify what are these harmful pathogens that are out there, and we can develop some species specific type testing to make sure that it's clean of that and sort of let the beneficial stuff go through, obviously within reason. Um,
Yeah. Yeah, and I
um,
think the problem is it adds an extra burden on the companies that are testing, because it's so much easier to just say, oh yeah, 10 to the 4 or 10 to the 3 colony
right.
forming units. When you have to start distinguishing between them, you need the PCR based type assays that you guys are developing to say, okay, it's a trichoderma, or it's a penicillium, or it's an aspergillus, or it's something in between. And I think that's going to be really important.
And so I imagine that sort of the goal would be eventually to maybe breed some varieties that have resistance to some of these pathogens, whether it be Fusarium or powdery mildew or things like that. And in general, how long does it take to sort of do that, first of all? And then second, do we even really have a full picture of all the pathogens that can potentially infect the plant?
Yeah, so with cannabis, I think breeding for resistance is quite doable, and a couple of reasons for that. Firstly, there's so much genetic diversity that I think it's not so much can we find it, it's how are we going to find it. I think the first step is going to be developing methods for screening. So whether you're screening for Fusarium or if you're screening for powdery mildew resistance, we have to have some really good methods developed that are actually already developed in other crops. So in tomato again, in cotton or basil, they've already developed methods for screening for Fusarium and powdery mildew resistance. So we need to adapt those methods for cannabis, and then go out and do a major collection, and maybe collect about 100 of these different strains or genotypes, and then run these methods through and try to identify, let's say 5 or 10 that show some level of resistance to either Fusarium or powdery mildew. And then I think it's a matter of making the crosses. So you induce some male flowers, collect the pollen, make the crosses like most breeders or most growers would, collect that seed, and then run the test again in the next generation to find those plants that may have picked up the resistance. So I think, after 3 or 4 rounds of this testing, we could identify potentially certain strains or genotypes that have resistance.
Yeah, and then second part of that question, do we even really have a full picture of the full breadth of pathogens that can affect cannabis?
Yeah, so I go out to I go out to the licensed growers about once a week. Usually, I go out on Wednesday, so that's my cannabis day. And yeah, I'm not surprised where we stumble across new diseases. And I'm not going to say there's like hundreds of them. I would say we've probably found maybe 10 or 15. And that's not to say that that's the maximum number of diseases we've found. There's going to be more. So you talk to the hemp growers in the U.S., they're always finding new diseases. There's Diseases coming from a different crop. There's diseases coming from a different area. There may be diseases coming from a windstorm or rainstorm that brings spores in from nearby areas. At some point, we are going to max out. We'll probably have a full list of diseases that are out there. But I think we've got the major ones. The major ones being powdery mildew, botrytis, Fusarium, Pythium, a number of leaf spotting diseases that are found back east, and then of course a couple of virus diseases that we haven't quite worked on yet. So I think the list is getting closer to being completed. At least I hope it is.
Yeah, add it to the list of things in the cannabis industry that we don't quite have figured out yet.
Yeah, yeah.
Um, yeah, and I did want to shift gears a little bit because I know that your presentation at this year's CanMed isn't actually about plant pathogens. It's actually about the development of cannabis trichomes. So I want to give you an opportunity to talk a bit about that and give us a little bit of a preview of what that's going to be about.
Okay, yeah, and I'm sorry this didn't work out for you guys this year to have the conference in Pasadena, but hopefully next year when COVID gets a little bit easier to deal with. Yeah, I was going to talk about some of the work we've done with trichome development and trichome analysis. So what we've done is there's a standard large piece of equipment called a scanning electron microscope, and what that allows you to do is actually magnify the bracts and the other tissues surrounding a cannabis flower, where you can magnify, let's say, 10,000 times, and really get in there and start looking at these developing trichomes. And we found two or three really interesting things. The first would be that we've found four different trichome types, and maybe I'll come back to that in a minute. There's at least four different types. The ones that we're all familiar with, of course, are the stalked glandular trichomes. Those are the ones that produce most of the cannabinoids. So we found the different types. We also found that there appears to be changes in numbers in those four as the plant matures. And so, again, as you probably would expect, more of the glandular stalk trichomes are being seen as the flowers tend to mature, because they're the sort of the horsepower behind producing all the cannabinoids. And then the other thing we did see is some differences between varieties or between genotypes. And so the excitement there is that if you could correlate those different trichome types or different trichome numbers to final THC, CBD, and other cannabinoid ratios, then that would be a really powerful way to say, okay, I'm going to use, well, maybe not a scanning microscope, but I'm going to use a way to magnify those flowers and look at them and see how many trichome types do I have and how many total numbers of trichomes do I have. And is that going to be a way for me to tell whether I've got 12% THC or 20% THC or not? So that's sort of where we're going with this study is trying to correlate what we're seeing visually with some biochemical analysis of those flowers.
So that's interesting. Is it possible that the different trichome types could actually be producing different cannabinoids?
So that's a really interesting question, and so let me just tell you sort of what those four types are. The first type is sort of the hair-like projections, the stachylids as we call them, which really don't produce any cannabinoids at all. They have a protective function. They're the ones that, these humongous hair-like structures that if I was an insect and I stumble into them, it's going to be game over for me. So those definitely have a protective role. And then, as I mentioned, is the stalked glandular trichomes, the ones that produce most of the cannabinoids. And then there's a shorter form of those, which are what we call the sessile glandular. And the sessile are pretty much the same as the glandular, except they have way lower levels of cannabinoids. In other words, they're an immature form of the stalked glandular. So that's sort of the second type. The third type is what we call bulbous, and the bulbous are tiny. They're so small that you could never see them even with a magnifying glass. You really have to have a high power microscope to see those. And so I don't know what those are doing in terms of their function. Are they producing cannabinoids? Are they producing terpenes? Maybe I don't know. And then the fourth type is what we call the peltate, and those are sort of like half moon shaped. In the microscope, they're half moon shaped, They're like saucers, upside down saucers, sitting on the surface of a leaf. They don't have a stalk. They're pretty large. You can see them. And again, I don't know what the function of those are. It may be terpenes, it may be cannabinoids.
That's really interesting. And then I thought reading through the abstract, it looked like you had looked at two different types or two different cultivars. Is it possible that if you were to survey more, you might even find other types of trichomes?
Yeah, it's possible. So we picked those two because they differed quite a bit in the THC content. So one was like 10% and the other was like around 18. And I think we saw that in the 18%, the higher THC CBD type strains, they were larger numbers of trichomes, and the stalks on those trichomes were much longer. In other words, they were the type of strain that you looked at with a hand lens, and you could see the trichomes really clearly, and you could differentiate whether or not they were sort of the milky type or whether they were the sort of the amber colored type. So at least for those trichomes, we think there's a correlation between the final THC content and trichome numbers.
Excellent. Well, I look forward to seeing those pictures. If they're anything like the photos that you presented at CanMed 2018 with the close up of powdery mildew and things like that infecting the plant, it's pretty amazing stuff to look at. I encourage anyone to go back and view the video of Zamir's presentation at 2018 because that was certainly a hit.
Yeah, I tell you, Ben, these pictures are way better. I mean, I'm really excited next year to try to present those because I look at them every once in a while and I think I'm like I'm in outer space. I'm sitting on the moon somewhere, and these huge projectiles because they're massive in the microscope. The scanning, they're huge and they're amazing. They're just an amazing sight.
Well, I'm looking forward to it. I think that's a great place to stop. Thanks again, Zamir, for taking the time to talk with us, and I look forward to seeing you out in Pasadena this spring.
Thanks a lot, Ben, and I appreciate all the work you guys are doing and hosting all these CannMed conferences. They're a great way to meet people. They're a great way to socialize, and I'm sorry we couldn't have done it this year.
It'll just be bigger and better next year.
Okay, thanks a lot.
I hope you enjoyed my conversation with Dr. Zamir Punja. Check out the links in the show description to learn more about the topics we discussed. And thanks again to our sponsors, the Growers Network and the Hemp Coffee Exchange. Our next episode will drop September 16th. In the meantime, please go to canmedevents.com/coffee talk and sign up for email updates. That will enter you into that drawing for 2 tickets to our CanMed 2021 VIP dinner and also keep you up to date with all things CanMed 2021. If social media is more your thing, you can find us on Facebook, Instagram, Twitter, and LinkedIn. Just search for CanMed Events. And lastly, if you are listening via a podcast app, go ahead and hit the subscribe button so new episodes will automatically download to your device. And if you'd like to leave us a 5-star review, we'd really appreciate that as well. All right, that's it from us. Until next time, stay safe, stay healthy, and be sure to come back for the next episode of CanMed Coffee Talk.